It leads us to the other challenge that I've also been focused on in the last decade, which is if we're trying to optimize a wastewater treatment system without building more tanks, without increasing sludgeage, and we're going out to bring microbes that are not natural or native to the system through bioremediation. What it means is that we are trying to introduce into that process microbes that natural selection does not prefer. Because if those microbes were if the natural tendency was for those microbes to grow, we would not need to bioremediate. They would already be in the system. We would have optimized for them. So anytime we're doing bioremediation, it means we're trying to introduce into a system microbes that nature has not selected for, and it poses 4 problems. The first problem is that any microbial system we go into has acclimated microbes. There are, it has the bacteria themselves, it has viruses, phagal organisms, phages, viruses that are native to the system that the microbes must be adapted to. And then it has other organisms. So the first problem we have is if we introduce these new microbes, one possibility is they get killed by things that are smaller than them, by viruses. The second problem we have is if we introduce these microbes new to the system, because we're trying to enhance the process, microbes are in the region of 0.5 to 1 micron in size. And a healthy microbial ecosystem would have things like rotifers, it would have water bears, it would have amoeba, paramecia, organisms that are 50, 100 microns in size that feed on the bacterial cells. So if we're introducing these bacterial cells, these minute, free-floating organisms that have not formed flock and are not native to the system, if they survive things that are smaller than them, like viruses, well, they may not survive things that are bigger than them because they might get eaten up because they're not part of the ecosystem. They've not formed flock. Bacteria in wastewater systems tend to form flock. So in a typical wastewater system, if you looked under the microscope, we're not seeing 1 micron organisms floating around most times. We're actually seeing these organisms congregate in flock of about 50 to 100 microns. In fact, that's the only reason why they can settle in a clarifier, is because they have formed this flock. They are protective. They allow them to intermix, to mingle, to be able to protect themselves in this polymeric matrix. But if we're doing bioremediation, then those microbes are not part of that ecosystem and they could be eaten up. The third challenge we have is if I do bioremediation, the average bacterial cell has the same specific gravity as water. So if I dose it as a liquid, the tendency is it just floats on the surface. And if it floats on the surface, I can only retain those microbes for the hydraulic retention time. However, in any biological treatment system, the solids retention time is always several orders of magnitude higher than the hydraulic retention time. So hydraulic retention time for most systems is in the order of hours. The solids retention time for many systems is in the order of days, sometimes weeks, sometimes even months. The only situations where HRT and SRT tend to be the same is in digesters, where you're not separating solids, you're just putting the solids into the digester, you're mixing the whole thing, and you need about 20, 30 days overall because it takes time for those hydrolysis steps to occur. So how do I make sure, if I was doing bioremediation and I'm bringing all these expensive microbes into a system, how do I make sure they don't just float away? So that's another challenge to deal with. And then the fourth one is, how do I make sure that when I introduce these microbes that are supposed to enhance the system, we can get them integrated into flock within the time frame that we have for the hydraulic retention time. The way that so this led me to my work in Drylet, and what Drylet did was, rather than just dose microbes directly into a wastewater treatment system for remediation, they would actually just embed the microbes inside a porous media. So the porous media would be some type of metallic oxide, highly adsorptive, and normally food-grade material, so things like silica oxide, and the microbes would be embedded. And the advantage that that provided was by protecting the microbes inside the media, you are protecting it from things like phages and predation organisms. So these media are about, typically 100 to 600 microns in size, so much bigger than some of the things that want to feed on the microbes. That protects them. The other thing is because the microbes are inside of the media in this biofilm environment, it becomes very difficult they're very resilient. I think the best example of biofilm resiliency I can imagine is the fact that we brush our teeth every day, and yet we can't kill the microbes that are in our mouth because they are in a biofilm form. And a biofilm form also offers remarkable advantages. Now we know there's something called quorum sensing. Now we know that there's lateral gene transfer within a biofilm environment. Now we are beginning to understand why all it takes in a biofilm is for one or a few microbes to survive and adapt, and then pass those characteristics and qualities across the entire biofilm environment. So that provides that type of protection. And then the other one is, in this metallic oxide, this stuff doesn't float. It actually, if you have a good mixing, it stays mixed. If the mixing is poor, it falls into the solid phase. So you retain these microbes in this form for the sludge age, for the solids retention time and not the hydraulic retention time, which allows you to keep the microbes within the system for much longer. And then now that the microbes are within the solid phase for much longer, you're able to integrate much better into flock. So as I indicated earlier, a number of different things to consider when we're looking at microbes. There's a dizzying array of them. There's so many of them we could potentially be bringing in, but there are just so many conditions we have to think about. So the first one is, the pH. Depending on the conditions that we're dealing with, what types of microbes are best optimized for the type of work that we're trying to do. Temperature is also critical. And so in the most resilient systems that I've seen, even if you would see microbes across a broad range of temperature ranges. And as you can see from some of these diagrams, even though the microbes might have optima in specific, so the mesophiles might have a 39 degrees Celsius optimum, but then there's a significant overlap at some point between psychrophiles and mesophiles. So we're typically not surprised when we do genomic analysis and we find psychrophiles even in mesophilic systems, because there is some overlap. It provides us with resiliency. It's one of the reasons why, winter comes and our entire system doesn't die off, because as soon as winter comes, it just means that now our population dynamics begins to shift a little bit. And with a keen knowledge of what's going on in the system, we can actually then begin to use things like bioremediation and process modeling to help us optimize what's going on in the system. Another key thing that that I've found extremely important in the work that I've done over the last few decades is what is it that we're trying to achieve? And when I started out, my interest was in the dissolved oxygen level. I've since found that it's not a helpful parameter. What's more important is, what is the ORP? What is the oxidative reduction potential of the system? Because the ORP, as we can see in the diagram below, gives us a good understanding of what the electron acceptor is likely to be in that environment. There are very many different types of electron acceptors. Carbonaceous organics can be electron acceptors, sulfates, nitrites, nitrates, oxygen itself. And so once we start going into things like nitrification, denitrification, we have a keen understanding that we're trying to stay around this anoxic zone, because if we're not in the anoxic zone, if the conditions are not anoxic, then our microbes are never going to utilize nitrates as an electron acceptor. They would always prefer to go to the materials of a higher ORP, that ORP environment. So if I want to do denitrification and I want to remove nitrogen, then I know that I must force my system into an anoxic zone. If I'm trying to do things like, anaerobic digestion, then I know that my ORP as a control element has to be more reduced. And so, the ORP as a control variable is something that I've learned, has even more importance than the oxygenation state of the process. A DO of 0 might be an ORP of 0. A DO of 0 is also an ORP of minus 400. You have no idea what the ORP is if all you're doing is tracking what the oxygenation state is. But by understanding the ORP, we can actually choose and select exactly what type of dynamic and what type of electron acceptors we're utilizing in the process. And so in the work that I've done, we've worked across very many different types of environments. We've done work in soil with oil spills and had to degrade hydrocarbon materials. We've worked in large lagoon systems, industrial systems, where, as you can see in the second image, you just have this is a rendering plant where they're dealing with slaughtered animal waste. And there's just a huge amount of fat oils and grease that just sits on these basins. And what you're seeing here is one of my colleagues actually spraying that biocatalyst material in order to be able to break down that high gunk organic material. And the image that I have showed below is what it looked like when that spraying began, and what that system looked like about 8 weeks later, where the microbes that you're spraying over the material is actually enabling you to begin to break down the lipophilic and hydrolytic, they're generating lipophilic and hydrophilic enzymes that are breaking down those solids and allowing the system to be, to essentially have an enhanced capacity for treatment. Sometimes we dose directly into the basin. We do a lot of work with lagoons, lakes, rivers, streams, septic systems, home-based septic systems, just wherever microbes are being utilized. One of the things that I've learned is we can actually understand, as soon as we understand what's going on in that system, without engineering anything else, there's a possibility that we can actually get treatment enhanced. So there's a broad range of things that bioremediation could be applied for. Traditional wastewater treatment system, improving performance in wastewater treatment systems, as you see in case 2, increasing biogas generation, whether in the context of large digesters, small digesters, farm-based digesters, mixed waste digesters, small septic systems in people's homes. It doesn't matter what the system is, as long as we're using microbes, they can possibly be enhanced. And then we also have also done a lot of work in concentrated animal feed operations. Dairy farms, swine farms, hog farms, things like that, where you're generating a significant amount of agricultural waste, both manure as well as crop residue, and you're trying to break that down because you got to do something with those solids. And then one of the other interesting applications is things like remediating algae. So anytime we see algae, it's a competition for nutrients. So we have nitrogen, we have phosphorus. The good news for algae is they don't need organic carbon. As long as there's inorganic carbon, there's alkalinity and CO2, the algae will grow. So if you're trying to knock algae out, then it's either you make sure there's no nitrogen and phosphorus, which can be very difficult because it means now you need treatment processes that remove nitrogen and phosphorus, or you stop runoff, or you introduce microbes that will compete with the algae for the nitrogen and phosphorus. And that's the approach in this particular instance. In case number 6 might be a little difficult to see, where the pond, a lake that was being used by divers, was overrun by algae. And the strategy here was to go in and outcompete the algae by introducing microbes that could compete for the nitrogen and phosphorus within the system. So one of the things that I've seen happen a lot more in the last, certainly in the last decade, is when we think about bioremediation, I am seeing, so when it comes to bioaugmentation, the work that I've done has mostly been focused on natural cellular bioaugmentation. So we've gone back into nature. We've looked at microbes that are naturally in water and in soil, and then we've taken those microbes that we know have some specific functions, and we've We've incorporated them into things like that metallic oxide platform, put them into systems. But what we're starting to see now is we're starting to see a lot of genetic bioaugmentation. We're seeing companies like Alonia and LanzaTech and Ginkgo Bioworks actually look at molecules they want to treat and then engineer microbes that can help, that can treat those systems. I have a bias for natural systems. I think there's a limitation. I am always wary of introducing new things into the natural environment. But this is just an area of work, of more work that I'm starting to see happen now. And then what I'm seeing a lot more, and I'm also starting to do some work in this space, is a lot of my focus historically had been on using microbes and having those microbes actually generate the enzymes that you need. But then there are companies that actually isolate enzymes. And you can also, in the same way that you're introducing the microbes, introduce some of these enzymic elements into processes to enhance treatment.